Hello, everybody. So we are going to be working again on the modeling meiosis and mitosis slides. So please go ahead and find them in your Google Drive. You should have saved them yesterday. If you did not save them yet, then you're going to have to start all over from the beginning. And you can find a blank copy of the slides in yesterday's folder for Monday. If you have not gotten started on this, please go back to yesterday's folder and follow along with the video and get caught up. So if you have done the Monday lesson and you're ready for today's lesson, go to My Drive. You might be able to find the slides right up here at the top. If so, just go ahead and click it and open it. Otherwise, find your class, so 8A Science most likely, or wherever you saved it to. Open up the folder, and then just open up your slides. And you should come back to this, and you should see that you have already gone through all of these because you paid attention and you followed along. So all of these cells should be filled out, and we should be on questions number 11 through 19. If I had a chance to finish this in Zoom with you on Monday, then you can go ahead and skip these next couple minutes straight up to the next slide. But for those of you who have not gotten this yet, let's go through it here. So Part C right here, a diploid set of human chromosomes contains 23 homologous pairs, or 46 chromosomes. We're going to fill in this table with the information regarding human body cells. So I'm adding to mine right here each human nucleus, and you can add that into yours as well. It just helps to understand a little more what we're talking about. Okay, so step interphase. How many cells are we paying attention to when we're looking at the cell cycle? Are we looking at all million bajillion cells in the body? Probably not. That's a bit much. We're going to be looking at just one cell. So type a 1 in there. So after mitosis happens, after everything is duplicated and moves to each side of the cell and splits up into cytokinesis, how many cells do we have then? So that's at the end of the mitosis. After everything's done, it splits up into 2 cells. That's the point of cytokinesis. All right, so the number of chromosomes in each human nucleus. How many chromosomes are there in one human cell? Let's reread this information up here. It says 23 homologous pairs, or 46 chromosomes. This is talking about one human cell. So if you take a skin cell, an eye cell, a cell from your lungs, a cell from your kidney, they're going to find how many chromosomes in there. There's going to be 46. Okay, so this is at interphase before mitosis starts. So once we get into prophase, it's going to double. It's going to come up to 92. And then all those chromosomes are going to line up in the center in metaphase, and then they're going to pull out to the sides during anaphase. And then they're going to move into 2 separate nuclei during telophase. And then at cytokinesis, they're going to split back up. So those 92 chromosomes come back down to 46. So the number of chromosomes before mitosis is exactly the same as the number of chromosomes after mitosis. So the last part, the number of homologous pairs, and remember, a homologous pair is a pair of 2 chromosomes that are exactly the same type. They may not have all the same information. If you remember Study Island, we talked about genes and DNA. So they might have different genes on it, but it calls for the same type of genes. So your homologous pairs will be the 2 chromosomes that work together to make your eye color, your hair color, something like that. And it says right here, there are 23 homologous pairs in a human cell, which means at interphase, there's 23. And at cytokinesis, it's the same number. It's still 23. So question 18, why is it necessary to double the amount of genetic material before mitosis begins? Go ahead and pause this video and think about that for a moment. Okay, hopefully you came up with some sort of answer that talks about maybe you need to have enough material to duplicate so that when you split, there's still enough left over. Maybe you said something about daughter cells. Maybe you said something about just the fact that you can't start with one and then split it in half and expect to have the same amount. You have to double it before you divide it. Question number 19. The two daughter cells end up with an exact copy of the genetic material from the parent cell. How does your simulation support this statement? So our simulation, if you're using Go Formative, was to actually use those pipe cleaners or that thread or that yarn or those papers, whatever you used to represent your chromosomes, that you moved all the way through all the pieces of the poster board and the diagram, that we moved from this stage to this stage, here, 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 and here. So if you did not use Go Formative and you only have these slides, then this is your simulation. So in our simulation, how did we show that the two daughter cells, so this one and this one, how did we show that they end up with an exact copy of the genetic material from the parent cell? And how does our simulation support that? So look back at these, think of that question. You can pause this for as long as you need, and as soon as you've got an answer, go ahead and type it right there. I'm going to move on to the next slides. All right, so all of the classes should be at this point now. So you are going to use your lab materials on your poster to simulate mitosis at your desk. Or if you did not have access to the lab because you have not gotten 100% on the lab safety test and or you have not turned in your contract, then you can only use this right here. If you would prefer, you can also still go ahead and use the slides just because we're here and I'm showing you right now. So we're going to fill in the chart with the number of chromosomes as you complete each step. So right here I have text boxes with little blanks in it, and you can just type in whatever number you think it is. It's not 5, by the way. All right, so use this information to fill in the table on the next slide. So first off, we started out with 2n equals 8. So we're back to a fruit fly again, and its diploid number, its full chromosome amount, the amount of chromosomes that are in a fruit fly cell at any given moment when it's not having mitosis or meiosis happening, is going to be 8. So at this first stage right here, the chromosomes double. So I'm going to take my 8 and make them 16. So I've got 2, 4. So each of these x's counts as 2. They are a homologous pair. 2, 4, 6, 8, 10, 12, 14, 16. So how many chromosomes are here? 16. All right, so chromosomes double, we're going to have 16. So I'm going to take my chromosomes. I started out with 4 of each color. I'm just going to put them out here because if I put them inside this diagram area, it's going to try to move that. So let's put our 8 chromosomes to start. And so that was our starting amount. And then we doubled them. So we've got another set right here. Let's go like that. And another set right here. So this is like right before prophase, kind of as prophase is starting, is when everything doubles up and we're going to get to our 16 chromosomes. So prophase is going to start. So I'm actually going to just grab all these and I'm going to move them down into prophase. How many chromosomes do you have? You still have that double amount, so we have 16. Okay, next we're going to have metaphase. So I'm going to move these aside just because they're kind of in the way right now and I don't need them. Because now our chromosomes are turned into X's. And they're going to be in metaphase, which is where they make a straight line all the way across that cell. How many chromosomes are there? Well, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So even though they're X's and I can pull them together, they still count as 2 chromosomes. They're still a homologous pair. So type in your number here. All right, so at metaphase, they're going to line up along the center. But anaphase, instead of these chromosome pairs being ripped apart, they're going to stay attached. But they are still going to go to separate sides. So I'm going to move this black pair over here. And I'm going to keep the yellow one of that same size. I'm going to keep it over on the right side. Then I'm going to grab one of these like second sizes and move it over. It doesn't matter if I move the yellow or the black one this time, just as long as at least one of them is different. Then I'm going to pick another. Let's go over there. And its equal size is going to come here. And then I can choose if I want to put the littlest one, yellow or black on either side. No, I'm going to go like that. This is a completely random organization here. And it doesn't quite matter if we're putting the information from this cell's parents, from its father or its mother, on which side. It just matters that I'm not having copies of the same set. So I can't put my biggest X's both in the same cell. They have to go to different sides. So that's anaphase in meiosis. It's different from mitosis because we keep them in their homologous pairs. We keep them in their chromatid attached. So as we go into end of meiosis 1, we're going to have these cells, right, or these chromosomes here, are just going to move down into that cell, and these ones are going to move down into this cell. How many chromosomes are in each of these cells? Well, let's see. I've got 1, 2, 3, 4, 5, 6, 7, 8. And how many are in the other side? 1, 2, 3, 4, 5, 6, 7, 8. All right, so metaphase 2. What's going to happen this time is our remaining chromosomes, they are going to go ahead and line up in the center. So, well, I've already got them kind of lined up, so I'm just going to move them down. Move them down. All right, and then so how many chromosomes do you have? We didn't change anything, did we? So we're still that same number. Last but not least, we are finally going to split up our remaining chromosomes into our gametes. So I can't pull these apart like we were able to do earlier, so I'm going to have to use these little sticks over here. So I have one of the big ones. So it's got one big and one big. And then I've got one of these kind of mid-sizes. So I'm going to go 1 and 1. And we're just going to stick one of each of these into our 2 little cells here. But I'm only sticking with the ones here on the left because that's what I'm working with. And I've got all the little sizes of the yellow. So there, there, and there. And there. And there. So if you chose a different arrangement up here at your anaphase, make sure to match with that. So I had my bigger X chromosomes over here in the black, and then all the smaller ones I made yellow. So I have it matching that in my little chromosome sticks. So over here, the 2 largest yellows, they're going to split, 1 to each cell here. And then the rest are going to be with these black ones. So I've got 3, and I've got 3. Okay, so now that we're at the end of meiosis, how many chromosomes are in each one of these tiny little cells. You can go ahead and type those in the text box, unless you did like I did and moved it over. Then you can just count 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. Go ahead and pause this slide if you need to, otherwise I am moving on. So questions number 20 through 31 on Go Formative. So when we started out meiosis, how many cells did we have? Were we looking at every single cell in the body of our organism, of our fruit fly, that can create gametes? No, that would be crazy to try to keep track of. We only looked at one cell. So start of meiosis is 1. How many cells did we have at the end of our first meiosis cycle? So our first mitosis, basically. Well, end of meiosis 1, we have 1, 2 cells. How many do we have at the end of meiosis 2? What is the end result, end of meiosis 2? How many cells do you have? We have 4. So we started with 1, and then it doubled, and then it doubled again. We have a double-double now. Okay, so the number of chromosomes in each cell. Go ahead and go back to the diagram and just follow through how many you had at each stage. So we started with our 8, which is our original cell. And then the information doubled, and then it split. How many chromosomes are in each cell after they split the first time? And then for end of meiosis 2, how many chromosomes are in each cell after it splits the last time? So I will ask you guys to fill those in yourself because you've already filled out that diagram. Okay. How many homologous pairs are in each nucleus? So unfortunately, I moved the sticks all the way down to the bottom, but up here, when we start out, how many homologous pairs were there? There was the one black with its matching yellow, another black and a matching yellow, another black and matching yellow, another black and matching yellow. There were 4 pairs when we started out. Okay, so each pair was 2 sticks of the same size. So I'm going to grab a homologous pair from over here, cut. So a homologous pair is 2 chromosomes of the same type. So they're both this biggest, come on, let me grab right there. So these right here are the 2 biggest chromosomes in my set. They make 1 homologous pair. So at the beginning of meiosis, there were 4 homologous pairs in each nucleus. How many are there at the end of meiosis 1? Well, every one of these little X's is 1 homologous pair. And we had how many at the end of meiosis 1? We had 1, 2, 3, 4 homologous pairs at the end of meiosis 1. So that's 4. At the end of meiosis 2, how many homologous pairs are there? So I'm going to go ahead and highlight those. And I'm just going to copy one set over. And I'm actually going to toss it into my table. Okay, so if you look very carefully at these chromosome sticks, do I have any that are the same size? How many of these sticks are the same exact size? Are there any? I don't see any. So if there's no sticks the same size, that means we don't have any homologous pairs. We don't have any pairs of the same type of chromosome. So what number should that be if we don't have any pairs? All right. So I'm going to leave a question mark because I gave you that answer. I just didn't tell you the number. So I want to make sure that you're using your brain and not mine. All right, so diploid or haploid number. Remember, our diploid number is the total number of chromosomes. So let me go ahead and do a text box here. Diploid equals total number of chromosomes in a cell, in a body cell. There we go. Your haploid number is half of that, or the number of chromosomes in a gamete. Okay, so diploid, again, is the total number of chromosomes in a body cell. At the start of meiosis, we have a body cell. So it's starting out with 8 chromosomes. So I'm going to say diploid. And that is represented by 2N. At the end of meiosis 1, how many chromosomes do we have? Do we have the full set or do we have a half set? So if we have a full set, then we're going to call it 2N. If we have a half set or a haploid set, then we only have N. I'm going to leave that up to you to figure out what we have. So once you figure out how many chromosomes are in that cell, you can figure out if we have a diploid or a haploid. And when you figure out how many chromosomes are in this final cells, in the final 4 cells here, then you can figure out if those are diploid or haploids and fill those in. And so let's see, I'm going to move on to the next slide. So what happens to the homologous pairs of chromosomes during meiosis 1? So remember, meiosis 1 is when we go from our starting point, our interphase, we go through prophase, and we duplicate this original cell into 2 new cells that have not separated out their chromosomes. So give me an answer of what happens to the homologous pairs of chromosomes during meiosis 1. And then you can also answer this on Go Formative. It gives you a multiple choice option. Question number 33. At the end of meiosis 1, how does the number of chromosomes in each cell compare to the original number of chromosomes in the parent cell? Is it a diploid or a haploid? So go back to the table, see what you filled out, and then decide, is it still 8, which would be a diploid, or is it down to 4, which would be half of that, or a haploid? So give me an answer there. Number 34. At the end of mitosis 2, how does the number of chromosomes in each new cell compare to the original number of chromosomes in the parent cell? So at the very, very end of this meiosis process, that should say meiosis 2. Please make that correction on yours. At the end of meiosis 2, how does the number of chromosomes in each new cell compare to the original number of chromosomes in the parent cell? And why did I put that diagram? That is the wrong one. My bad. All right, so at the end of meiosis 2, we have our gametes. Are those diploids or haploids? And last but not least, the last couple questions. So right here, we have fruit fly sperm and fruit fly egg, and 2 questions about it. So I went ahead and split up the information into 4 chromosomes. And so this is 2 chromosomes from the father, 2 chromosomes from the mother of the male fruit fly here. And then in this case, I have maybe 2 chromosomes from mom, 2 chromosomes from dad of the female fruit fly. They are going to meet together and form a zygote. And so we're going to take all these chromosomes here, and I'm going to form my homologous pairs. Oops, those are different sizes. There we go. Those are the same size. Those go together, and these go together. Okay, so I now have my 4 new homologous pairs, and when they join together, by taking 4 chromosomes from the father, 4 chromosomes from the mother, it makes 1, 2, 3, 4, 5, 6, 7, 8 chromosomes in the child, or in the zygote. And so we have a mixture of mom and dad's informations and grandmother and grandfather's information are all right here into the zygote. So your final chromosomes is how many? Count 1, 2, 3, 4, 5, 6, 7, 8. And do we now have a diploid or a haploid? And that's all you guys need to do today. If you have not gotten 100% on your lab safety test, please make sure to take care of that today if possible.